Hello everyone. Yes, welcome to the askIITians. I'm your Sumit Marik, biology faculty, and here we are going to talk about a very important topics. That is, rather than topics, I will tell you I'm going to cover like a marathon, like a genetics and evolution. Most of the students has faced issue in genetics and evolution, right? So this power pack webinar is presenting in a such a way that you will clear the doubt. See, this is the marathon, right? So we have to go through each and every topic very clearly, right? That is the thing. Any doubts and any something you can interactive online, right? So we are going to start that webinar on the genetics and the evolution. I know, guys, both the chapters are absolutely vast, right? Yes or no? And you have faced some issue while studying it. So here, for a purpose of the best revision over here, we are going to start a webinar or one short webinar, rather I can say that, right? On genetics, evolution. Okay, so let's start it. So everybody, those who are live, those who are listening to me, please give me a response that yes, you are there and you are listening me. Yes, it will be the quick, enthusiastic interaction between us guys, right? So, can we start? Am I audible to all? Let me know, everyone, please. Yes, am I audible to all? Yes, I'm getting a lot of response in the chat box. That's great. That's great. Okay, so let's start without wasting a single second over here. We're going to talk about again, I'm saying genetics and evolution. So all are get ready with your things, right? So here we start, and absolutely you'll get a notes after this thing. This notes I specially made for the quick revision. Okay, thank you for responding in the chat box. Thank you so much. Now, as I just inform you, this has to be the interactive session, right? So I want to extend the energy of mine and yours. Okay, so here let's start with a great enthusiasm, that is genetics and evolution. So, firstly, we'll talk about the principles of inheritance and variation. See, the genetic unit has two things, principle and the molecular basis. The principle, mostly we have to deal with the Mendelian inheritance. Yes. And you know what? That person, the scientist is called as the father of genetics. That is the Gregor Mendel, known as father of genetics, conducted the experiment. So he has conducted the experiment on which plant? Pea plant. Can anybody tell me why he choose only the pea plant? There are three criteria for that. Anybody in the chat box or you want to unmute, you can talk about that. Why only the pea plant he selected? Tell me, tell me, tell me. Yes, I'm waiting for it. Why only the pea plant? I want that interaction. Can I say that? Because of the short lifespan of the pea plant and the yes, exactly. I'm getting the answer. That's great. They are short living cycle, fast reproducing cycle. Very good, very good, very good. So yes, for this reason, Mendel has preferred only the pea plant because it's very easy to do the experiment. It's a short life and definitely the pollination can be done. Self pollination can be avoided over there, right? Okay. Now, as we know that the Mendel has given the laws. So what are the important points? I'm going to cover it what. Okay, so let's go. Let's start with that. First is law of dominance. What is that law of dominance? In a heterozygous condition, always remember it's a heterozygous condition when you talk about the law of dominance. The dominant allele, like you know what, if I'm talking about T and T, capital T is always dominant rather than this T. So this one is a dominant, this one is a recessive, right? So the one which is getting expressed in a F1 generation that states the law of dominance. That is what? It is a dominant allele. Exactly correct. Then we go for the law of segregation. What is law of segregation? The word itself says that purity of the gametes. Even gametes are together. For example, this capital T one and this small t is together. Okay, even they are together, but they are not mixing. That purity has to be maintained. So law of segregation states that each parent contributes only one allele for the trait. Trait means characteristics, right? So one character will be passed on to the offspring as allele pairs separate during the gamete formation. So what does it indicate? It indicates that whenever the two gamete combine, but actually they are not combined. They are still there together, right? They are still together without interacting with each other, without creating any problem to each other. That is a law of segregation. And the third, law of independent assortment. Yes, independent. What is that? Different genes assort independently during the formation of the gamete. See, guys, these are very, very important. Law of dominance, law of segregation, and law of independent assortment. Okay. Now, as a Mendel is called as the father of genetics, he gives these three laws. Now, what experiment he's performed on what basis? Okay, that we understood that, but he chosen the first one is a monohybrid cause. What do you mean by mono? Only the one. So it involves a single trait. See, instead of having like that, right? We are studying many subjects at one time. It's very difficult. So let's focus on one subject at one time. Similarly, that's Mendel also have done the same thing, right? Involves a single trait. He just work on the single trait characteristics. For example, let's take a height of the pea plant. Then the phenotypic ratio. What is the phenotype? The physical appearance. That is 3:1. So all are tall. Tell me one thing. If I'm writing TT, TT, and this TT, tell me, my dear students, when I'm talking about the first TT, always is dominance. It will be the tall. But don't you think so? This is heterozygous. Yes or no? But still, it's giving the effect, phenotypic effect as a tall because the dominant character is capital T, exactly correct. And one recessive is there. So this one and this two and this one is recessive. So 2 plus 1, 3:1 we ratio get it, right? Yes, everybody is with me? Yes, exactly. So this example they have given over here in a monohybrid cross. See, monohybrid cross is very easy to understand because only one character they have talk about. Now let's talk about the dihybrid cross. Whenever they took the two characters, now there is a combination of the 16. You know that, right? That chart. Do you remember from NCERT? See, NCERT, you have to understand all the diagrams and all. So from that diagram or from that thing, we know that ratio is 9:3:3:1, right? Sir, how come this much ratio? Because we have the 16 combination. From out of 16 combination, we have this thing cross between the yellow round and green wrinkle, we get the 9:3:3:1. So there are two characters. Can you see that? The two characters they have utilized. Okay. So let's proceed further. Now, majority of the students have this problem, or what we can say that they have something issue with this test cross and back cross. This question maybe arise in the CBSE board regarding what exactly test cross and back. So here the answer is when you talk about it, test cross, obviously test, we have to test something. So you have to remember like that, right? So cross between an individual of unknown genotype. When you're testing something that is unknown genotype, you have to use it, right? And homozygous recessive individual. Why you have to go for recessive individual? Anybody can from the online interaction tell me why only recessive individual is choosing in the test cross? Because yes, because we want to understand if there is a dominant characteristic of the unknown genotype, it will be give the phenotypic ratio or phenotypic appearance, so we can understand some gene is there, right? So for that, test cross is very, very important. Now, so what do you mean by back cross? So back cross is nothing but crossing with F1 generation, whatever the parents, insert the parents we are going to talk about, right? So here we talk about the test cross and the back cross in detail. So make sure you're reading this before you are going for the examination, right? Make sure. Now let's talk about the deviation from Mendelism. So there are also deviation. Yes, there are deviation. Everything is not exactly same what has a Mendel has suggested to us, right? So neither allele is completely dominant and the heterozygous phenotype is the intermediate between the both and parent. Sir, I'm not getting this. Okay, so I'm here to tell you. We are talking about the incomplete dominance. What do you mean incomplete dominance? See, the flower is indicating a pink color. Why there is a pink color? Suppose you're crossing between the red color and this another color, white color. Let's take example, white color. So now the incomplete dominance means what? The neither allele is completely dominant. Means either the dominant should have given the 100%, right? But it is not giving the 100% dominance. So both alleles showing the 50-50%, or what we can say, they are getting mixed. This incomplete dominance is also called as a mosaic one. Yes. Example, Snapdragon. Please remember this example is very, very important. Snapdragon flower is also called. Yes, exactly. This is a scientific name of that. So that is an incomplete dominance. Don't get confused between the incomplete dominance. Incomplete means the allele is not producing their 100% effect. So they will be incomplete. Like we have a red and white. So there is a combination. What will be the intermediate? Pink color flower will be there. Very good. Now let's talk about the codominance. What is the codominance? Co means what? Like I use the word colleague, right? What is colleague? Though both work together, right? So codominance, both are dominant over here. So both allele are equally expressed. If they are not equally expressed, they call N. Yes or not? The first one. Now we are talking about the codominance. Anybody has any doubt? You can ask in between. Both the alleles are equally expressed because it's a quick recap. So equally expressed is called a codominance. Now, sir, there is an example of the blood group AB. Why there is the example of blood group AB has given to this? Can anybody tell me? Yes. When you talk about the blood group, we have a ABO blood group system. Out of that, when you talk about the A and B, that means they are co-dominant. That means both are expressed. Yes or no? That's the reason the example is given for codominance of blood group system AB, where both A and B antigens are expressed. Okay. Now let's go for the next one. That is a deviation called as a multiple L. Sir, in codominance also you have taken the blood group, and here also there is a blood group. So what is the difference? Let's understand. When you talk about the codominance, that means both are expressed. It's a combination A and B, both are expressed. But when you talk about the multiple allelism, what is given that more than two alternative forms of the gene exist in a population that is indicated. So we have a B, and O system. So tell me one thing, those who have a antigen A will have a blood group A. Those antigen have a blood group, that antigen have a B, that is blood group B. And nil, that is blood group O will be there. But can you see that the ABO blood group system includes IA, IB, and I, that is nil. So three alleles for the one blood type inheritance. Don't you think so? More than two? Yes, that's why it's called multiple allelism. So there will be the question. See, in your board, they can ask you this kind of question. Justify the blood group is always is showing codominance as well as multiple allelism. So you have to explain this. In the codominance, both are the getting expressed. But in multiple allelism, more than two alternative forms are there. That's why it's called as a multiple allelism as well as codominance. This point is very much clear. Give me a thumbs up. Yes, students, give me a thumbs up for this. This is very, very important for this justification. Application-based question will be there. So have you understood that? Give me thumbs up for it. Yes, I'm waiting for it. Let's, yes, 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 yes. Very good. Thank you, Aditya. Now let's go to the next one. Now, there is a polygenic inheritance. See, again, I'm telling you, it's very difficult to cover all the points, but I will make sure I will touch all the important points. So here, polygenic inheritance. Poly means many, right? So multiple genes control a single trait. What is the example? Your skin color is the example. Human skin color is controlled by the multiple genes, and the height influenced by the many genes and the environmental factor. So that is polygenic inheritance. One multiple gene controlled by a single trait. Are you getting my point? That is effective. That's called polygenic. So what is the summary? Let's talk about incomplete. We are getting the example snapdragon because we get a pink flower. Why it's a pink? Because it's an intermediate. So it is called mosaic dominance as well. Then in codominance, both are getting expressed. Then multiple allelism, more than two alternative forms of the gene exist. And the polygenic, multiple gene control of the single trait. Example is skin color and the height. Okay, so we have covered this much. Now let's understand what is next. Now we have a beautiful chart over here. Why I made this table for you guys? Let's understand it. Because everywhere, when you read from the NCERT, see NCERT's Bible, right? For it's right for your NEET preparation as well as for everything, right? So I made one table regarding every information is in this table. So let's understand. When you talk about the monohybrid, we have this cross. What is the phenotypic ratio? 3:1. What is the genotypic ratio? And the example, height in a P plant. So I will suggest all the students when you do this table, it will be very, very helpful to you during the your study time, during the revision time. It's quick revision. Okay. So you can take a screenshot or you can make it by yourself as well, right? So make sure you're doing it. So then dihybrid, we have this ratio. Then genotypic ratio, 1:2:1:2, 4:2, 1:2:2:1. That big ratio is there. You also understand that. The example is given. Then incomplete dominance, codominance, multiple allelism, we have just covered the things, right? We have just covered the things regarding the same. Then there is a sex-linked inheritance. We're going to talk about that in detail, but I just put one point over there. Please pay attention. Sex-linked inheritance, we have a X-linked recessive. Example is hemophilia and color blindness. So you should know that it can become in MCQs as well. Hemophilia and color blindness are sex-linked. recessive disorders. Okay. Then linkage and crossing over, we have a linked genes does not follow 9:3:3:1 ratio. Depend on the distance between the gene. You know that, right? If two genes are very close together, if they are trying to undergo the replication, what going to happen over there? If the copy down is there, what going to happen? We can't make the change. Yes or no? We can't. They copy as it is. But if they have a adequate or sufficient distance in between that, they can actually copy. So that is a linkage one, right? You should understand that. That experiment is given by whom? Yes, anybody? Can anybody say in chat box? Let me see how much you know about it. Okay. Yes, yes. Okay, that is Morgan's, and he has done on Drosophila experiment. Okay, that is a linkage and crossing over. That is also important point. You should aware about. Then let's talk about the chromosomal basis of inheritance. Now, this theory is actually put forward by the Sutton and Bowlby's chromosomal theory. They have taken the help of Mendel, right? So genes are located on the chromosomes and their behavior during the meiosis explain inheritance. Try to understand why they are talking about meiosis, because the meiosis is the only process where the meiosis is the reduction division, where this will be separate, and that time only you have to study about it. So linkage and crossing over done by the Morgans on the Drosophila experiment. Link gene tend to be inherited together. Why? Because they have not sufficient distance in between them. So they have, as usual, they could get copied out. There is no crossing over happen. What is the crossing over? What is the crossing over? We already done in 11th standard as well. What is that? Leads to recombination, increasing the genetic variation. So whenever the organisms want genetic variation, definitely there should be crossing over. For the crossing over, we need that link genes should be present on the chromosome definitely, but distance should be more or adequate enough. That is called centimorgan distance. Yes, exactly. Someone tell me in a chat box. That's great. That's great. Yes. Now let's talk about this. See, this is a quick revision. So let's understand this. Tell me one thing. What is that? Sex determination system. We have to go through this. Very important. XX and XY system is there, right? In this case, X and XY system, it followed by humans and Drosophila. Males contain XY, females contain XX. Then XO. What is the system? Double X, XO system. O means there is what? It followed by the males. Don't get confused between that. Example is grasshopper, and female have the proper this XX. One chromosome is missing in the males. Then we have ZZ system where this is followed by the birds and the reptiles. The males are ZZ and the female are ZW. So, my dear students, don't get confused. See, X and XY, this is you about to understand very well. But you get confused between two systems. So, make sure you're doing this thoroughly. Have a look over there. And now, after that, we're going to talk about the haploidy. What is that? So bees and ants, you know what? Honeybees, they have different behavior. Why? Because the ultimate they have to collect the honey, so they need a drones majority and only one female which act like a queen, and she the only work is there for the female is reproduction. So they don't need the much more female. So according to this system, the males are haploid from the unfertilized egg and sperm. But whenever there is a egg is deployed from the fertilized egg, the female will be formed. That is the honeybee, right? Guys, do you know one important thing about the honeybee? There are multiple female will be formed, but they will argue in between. There will be competition in between the bees, the female bees, and the one who won that is declared as the yes queen of that honeybees, right? So that is the thing. Okay, so just have a look over there. Yes, I'm giving you 2 3 seconds. You can go through it very well. Okay, now we are moving to the next part. That is types of mutation. First of all, tell me what is a mutation? Sudden change, right? You know that word. Sudden change is called mutation. So at DNA level, we have a substitution. Like, so what is substitution? We are substituting something. Some deletion. One can be left out. Okay. Then insertion. We are adding something. Inversions. We can flip that codon system, right? Yes or no? So these are the types of mutations. So yes, mutations are very important. Yes. So what are the mutations? Here we go to understand this table. See, I always prefer table for the quick revision. It will be very helpful, and you're making it, then it will be very, very helpful to you, my dear students. Now, point mutation. When you talk about the point mutation, itself is saying single nucleotide change. One nucleotide is getting changed. Example is, but see, because of the one, there is a lot of change. Sickle cell anemia is going to form, right? You know that valine is getting over there. Yes or no? Yes. So mutation in the beta globin gene, so leads to the abnormal hemoglobin. So those who are suffering from the sickle cell anemia, they have abnormal hemoglobin shape. Then frame shift, insertion or deletion of the nucleotide. Just we have seen above, right? What is this? Insertion and deletion. One is getting deleted, one is getting adding over here. You can see that. Ideally it should be यहाँ present होना चाहिए था, but यहाँ पे present ही नहीं है. यहाँ पे क्या हो गया? एक ideally कुछ नहीं होना चाहिए था, but यहाँ पे add कर दिया एक nucleotide, right? So that makes the mutation over here in case of frame shift, right? You should remember this. It's okay if you're not remembering this. It's okay. Trisex, this is there for the friendship mutation. Okay, then we have a deletion, loss of the chromosome. Okay, see, this is not in NCERT, but I have written for your information, right? So deletion in the chromosome spike causing the intellectual disability, cat-like cry. Okay, so the person who has this chromosome 5, if it is there a deletion, the baby cry like a cat, right? That is called cry-do chat syndrome, right? Very interesting, right? Yes. Then we have a duplication, repeating a chromosome segment leading to the extra gene copy. Copy करते टाइम क्या हो गया? Extra copy हो गया, right? So that is a duplication of this gene. Okay, this is not in NCERT. Again, I'm repeating. This is for the information. Then inversion. What is that? Reversal of the chromosome segment. Inverse, okay, right? Hemophilia A is going to happen because of the factor A gene will be inverted. Then the translocation. What do you mean by translocation? Exchange of the segment between the non-homologous. Non-homologous means there is no sister chromatid is there. While copy down the information, they are non-homologous. Okay, not similar. So that will be the translocation will be happening. Okay, now what is the summary over here? Chromosomes carry the genes and control the inheritance, while the mutation create a genetic variation. So mutations are useful? Yes, they are useful because the genetic variation we can get it. Sometimes beneficial, but sometimes harmful. So both we are getting the things from there. Okay, so let's go further and let's talk about is genetic disorder. The first one is Mendelian disorder, a single gene mutation. That's why we have lung mutation, right? You should know this is a single gene mutation. First is hemophilia. It is X-linked recessive disorder. X-linked recessive disorder. Effect is what? Blood fails to clotting नहीं हो रहा है यहाँ पे, leading to the excessive bleeding. Hemophilia is very dangerous, right? Example, you know that Queen Victoria's family is suffering from the hemophilia. Yes, there is a genetic disorder they are having. Then we have a sickle cell anemia. It's a autosomal recessive disorder. What we have the effect? Abnormal hemoglobin. The RBC become crest shape. Ideal RBC should be like this. The function of RBC carry much more oxygen. So they devoid of the nucleus once they get mature, right? But if the shape is like this, then the function is altered, right? So it is very problematic. So high prevalence in the African population. Why there is a African population are always affected because of that? Because the environmental condition. What is the environmental condition? They are prone to malaria resistance. So the African people have this kind of RBC, but for us, it will be harmful. I told you, the mutation has 2 points. One is beneficial, one is problematic. So for African people, it is very useful because malaria resistance will be there. So that we talk about is sickle cell anemia. Now let's talk about the phenylketonuria. This is also very important. Abnormal recessive disorder. These are the recessive disorders. Body cannot break down the phenylalanine, leading to the brain damage if it's untreated. See, a lot of problem is there just because of the one change of the mutation, right? One change of the nucleotide. It requires spatial diet for prevent the mental retardation, right? Sometimes harmful, sometimes beneficial. See, we have understood that. Now, let's go to the next point. That is a chromosomal disorder due to the chromosome aberration. When you talk about a chromosomal disorder, okay, the first is Down syndrome. It's also called as the trisomy 21 because there is a problem at a 21 chromosomes. There is a triple copy at the 21 chromosomes happens, right? So what is the effect? Intellectual disability, short stature, ज़्यादा बड़े नहीं होंगे, short stature रहेंगे, and they have a heart defects. ठीक है, इनके दिल में problem हो सकता है. Now symptoms: flat facial features, slanted eyes, and delayed development. So they are mentally retarded. That is all about it. Down syndrome. Then we have a Turner syndrome. Can you see that? Ideally, it should be XX or XY. We are one is missing, right? So that is called monosomy X. Only X chromosome in female found. See, that is X, so dominant will be X only. So they are female, definitely. But they are short stature, and the problem is that they are infertile, underdeveloped ovaries will be there, and symptoms web neck, heart defects will be there, and they will not found the menstrual cycle because they are not complete, right? So they're infertile. Okay, this is the reason for it. And then next is Klinefelter syndrome. XX, okay, that is a female, and XY is a male. We are aware about that. But whenever there is a XX and Y combination, the problem is that it is XY, if you're considering, it is a male one, but what is that? Extra X chromosome leads to the more female characteristics. So there will be little dominance over here. So infertile, definitely because of extra X chromosome in males. Small testes will be there, low testosterone levels. What is the symptoms? Tall height, less body hair, and they have a gynecomastia. What is gynecomastia? The breast development found in the male. That is called gynecomastia because of obviously here the chromosome got altered, and because of that the hormones ideally should be in male or female. The different levels are there. If estrogen is more, it leads to this one. Okay, so that is the Klinefelter syndrome that we have talked about. Let's move the further, and let's talk about now molecular basis. So here we completed the one, that is inheritance, right? So we have completed the inheritance. I understand that for you guys this is a quick revision. Okay, so let's talk about now molecular basis. Before I go ahead or deep dive into the molecular basis of inheritance, we should understand in inheritance principles, we talk about the experiment, what will the effect, chromosomal theory. That is the important point we have done, right? Now, let's talk about a molecular basis. When you talk about the molecular basis, we actually talk about the DNA structures and all. So let's start with that. So again, I told you, I will touch the important points because we have to do the quick revision, recap for that. Now, DNA structure and packing. Who has invented that? Watson and Crick model of the DNA. That is very, very important question. In 1953, with the help of X-ray diffraction images, so they found that two anti-parallel strands, 5' to 3' and 3' to 5' twist around. So they are like this. They are twisted. They are twisted, right? So sugar phosphate backbone. What is a backbone? Sugar phosphate backbone. Now, my dear student, you should understand what is a nucleoside and nucleotide. Okay, you should well aware about that. So in with a nitrogen base pair inside, base pairing is what? A bonds with a T, G bonds with a C. So adenine and thymine will have a 2 hydrogen bond, and cytosine and guanine have a 3 hydrogen bond. Okay, so that is the bonding. When we talk about the DNA, we draw this bond. These are actually the hydrogen bonds. Okay, now this question must be appear in the board exam. What is the Chargaff rule? So it's proposed by the Erwin Chargaff, and what is exactly? It's very easy, right? So what is that? A always bond with a T with a 2 hydrogen bond. C always bond with a G with a 3 hydrogen bond. That we are aware about, right? So purine, that is A and G, we always have the thing with the pyrimidine T plus C. So the ratio varying among the species but constant within the species. So suppose I have a plus T. You know that, right? Let me talk about in a very simple manner. When you know that A is equal to T and T is equal to C. So can you found out this equation? Yes, exactly. Purines is equal to pyrimidine, species to species. So if I know A is 55, for example, then T also will be 55, right? So I get one value, right? So if I know these things, I also get these things, right? So this is the equational based thing. You can do it very easily, right? Now let's talk about the DNA versus RNA. DNA, deoxy, this is ribose, double-stranded DNA, this is a single-stranded, right? We have different bases. What is the different bases? Instead of thymine in DNA, we have in RNA, we have a uracil. That is the only difference over here. Stability, DNA is a more stable and RNA is a less stable, right? In NCERT, there is a one paragraph, RNA world, right? Why? Because it's a less stable, but some scientists prefer that RNA is the first thing. Okay. Then genetic material in most organisms, genetic material in some viruses helps in a protein synthesis, that is RNA. We know that, na? mRNA, tRNA, right? Yes, exactly. So this is the difference between DNA versus RNA. Now, what are the types of RNA? Just I mentioned mRNA, that is messenger RNA, carries the genetic code from the DNA to ribosome. So this is found inside the nucleus, right? DNA to ribosomes. Genetic code they will follow. Then we have a tRNA that is found in the cytoplasm, act like a transfer RNA, brings amino acid to the ribosomes as per the whatever the mRNA has transcribed. Then RNA, ribosomal RNA, structural and enzymatic role in the ribosomes. Ribosomes are protein factories, so they need this thing. Now, DNA packaging, we have to talk about what is that nucleosome model. What is nucleosome model? You must aware about this diagram, right? In NCERT also, this diagram is there. Yes or no? So tell me one thing. It starts with the DNA, then it's a winding over the bed-like structure is there, the chromatin fibers, the octamers over there. Yes or no? Then it become condensed and more condensed, right? So you should aware about that. In board, they can ask you regarding this questions also, one mark question. So what is that? Let's understand. Nucleosome. What is nucleosome? The DNA wrap around the histone octamer, that is called nucleosome. Then chromatin fiber, nucleosome further coil more, and chromosomes highly condensed. So we should aware about this diagram in detail. Then DNA replication, we know that Messelson and Stahl is the scientists, they actually talk about the DNA replication. It is a semi-conservative. They can ask you the reason as well. Why it's called a semi-conservative? So you have to write this. After one generation, hybrid. You know that the experiment is there. Two generation, they find 50-50%. So according to this experiment, they concluded that it is a semi-conservative. So each new DNA molecule consists of one parental. You know that one DNA strand is destroying one. A new will be forming. Yes or no? Here will be forming new, here will be forming new. Again, this pattern will be repeated, right? What are the enzymes involved in DNA replication? Let's quickly revise. Helicase, unwind the DNA helix. DNA polymerase, synthesize the new DNA strand. Primase, lays down the RNA primate. Don't forget about the primase. This is important. And ligase joins the Okazaki fragments. Where you find the Okazaki fragments? Anybody from the chat box? Tell me. From the online interaction, tell me. What are the Okazaki fragments and where do you find? On the lagging strand or a leading strand? Yes? Yes, exactly correct. So when you talk about the this Okazaki fragments, the question should be appear this leading strand and the lagging strand. The leading strand synthesizing 5 prime to 3 prime. Always remember 5 prime to 3 prime is the direction for a leading strand. And lagging, because of the lagging, the see primer will be there, but they can't add. RNA polymerase can't add from this 3 to 5 dash. So there will be formation of fragment. Can you see that here are fragments are found? That is called the lagging strand, and these fragments are called Okazaki fragment. Okay, so this is leading versus lagging strand. That is also important. Now, central dogma, you know that DNA will convert into the RNA, mRNA. Then mRNA goes to the ribosomes. Then there is a process happen, right? So ultimately, from DNA we get a protein. So ultimately, it is a central dogma which includes the transcription and the translation will be there. So in retrovirus exam HIV, RNA converts back to DNA using the reverse transcriptase. Please make sure you are understanding this reverse transcriptase in only virus. Now let's talk about the transcription. So translation and transcription, we get confused. Transcription, just to copy down information. And translation, whatever you copy down, mRNA has copied down, that has to be done into the out product as a protein, right? So let's talk about the transcription. My dear students, whenever you are under the stress, you don't read the proper question. Sometimes it's asked translation and you write all about the transcription. So make sure that you're reading it properly. Give me a thumbs up that this point you understood. Yes, don't be in hurry while reading transcription or translation. Okay, give me thumbs up that you understood this point. Yes, and promise me you won't do this mistake, right? Okay, great. Now let's talk about the transcription RNA synthesis. So what are the process? In prokaryotes, they are very chilled. Why they are very chilled? Because only single RNA polymerase is doing all the activity. The RNA polymerase act like a hero over here, right? But eukaryotic, there are some challenges because there is no one polymerase. We have a 1, 2, and 3, and they have a different functions. You can see that they have different functions. RNA polymerase 1 is devoted for the RNA synthesis. 2 is for mRNA. 3 is for tRNA, right? That is a different story over here. So tell me one thing, when you talk about the eukaryotic transcription, see what is going to happen over here? The chromosome, we have to copy down the information. That is not up till now mRNA proper. Why? Because it is undergo the splicing, then undergo the translation, and then it can, undergo some events like a splicing, and then it will be coming out from the nuclear pore, from the nucleus, and then it goes to ribosomes, right? So we have to follow some things. So what are the things? Capping at the fifth dash, there is a cap addition. Then we have a telling at poly A. How many units? 200 to 300 units are getting added over there. In splicing, removal of the introns. See, in DNA, we have that combination of intron and exon. Okay, so introns are the non-needed one, but the extrons has to be preserved with the help of intron. But while popping down, we have to remove the introns, so undergo the splicing, then the telling and capping. At the capping, methyl phosphate group is added. Please remember, methyl phosphate group is added for a capping at the fifth dash. And three dash, we have a triple, A adenine that helps in what? Coming out that mRNA from the nuclear pore. That is telling. Then see, that is a NCERT diagram over here. The introns are removed, undergo the splicing. Then we have a fifth that methyl gonoside phosphate is there on third. We have a adenine tail-like structure is there. 200, more than 200 units are there. Then difference between the transcription and prokaryotic, just we have discussed. Very easy, right? Occurs in the cytoplasm. This is occurs in a nucleus. Then for initiation, they have a sigma factor for a promoter region, right? But in case of eukaryotic, it is not happening. Why? Because multiple transcription factors are available over here. After that is processing of mRNA, no post-transcription required here. But in eukaryotic, capping, splicing, and telling that has to be under. So this is also a table that improves the understanding. Let's go further. Now, when you talk about the stability, you know that it's a short life, but mRNA has a longer lifespan in case of eukaryotic. Try to understand this, okay? Now, let's talk about the genetic code and the translation. Have you noticed, my dear students, I always use the table for the quick revision. It helps, right? So what is the genetic code? It's a universal, same in all organisms. Then degeneratable and the non-overlapping, right? So we have a 333 codon. So degenerate UCC, what is this? All code for the serine. So there are, see we have a 64, right? Why we have a 64? That reason is very interesting. You are aware about that. What is the reason? Because we have three combinations, right? So they are non-overlapping. Start codon and stop codons are found. In important codons, we should know AUG, that is for methanoin. Methanoin also act like a start codon. AUG also act like a start codon, and it's also codes for methanoin. Then we have a stop codon, UAA, UAG, and UGA. These are called nonsense codon or a stop codon. Okay, just look at it here. Now let's talk about the steps in translation. Now we're done with the transcription, right? Now it's a translation. Initiation, elongation, and termination. mRNA first binds, then elongation, then termination. So this is very, very important in a translation process. See, I have come up with this diagram. You always know this. I already know aware about this in a NCERT. So I'm just going quick. What is happening first? The tRNA brings the amino acid according to whatever the code it reads in this direction, right? Then second tRNA jump into the picture. Then third tRNA jump into the picture. And have you noticed methionine gate is adding to phenylalanine, then phenylalanine is getting added to this. So this protein, when they form, they're getting added, or there is a formation of long chain of the protein once the tRNA jumping into that. So initiation, elongation, and termination. Once the stop codon enter, this process will be stopped and everything will be, yes, it will be terminated, right? So these are the key points. So termination, that is also important here. Which codon? This codon. We have talked about that, right? Let's talk about the key player very fastly. tRNA carrying the amino acid. That is the first step. In peptide transferase, enzyme is responsible, and mRNA occurs. Codons guiding sequence. And once all the last codon comes, that is stop, then this process stops. Okay. Now gene regulation, a favorite, what we can say, the favorite question. Most of the times, this question they ask, keep on asking the same question. Operon concept, lac operon. So we have a switch on and switch off model of the operon. Okay, so that is the lac operon system where the lactose is present. That time only this get activated because lactose has to be break down into the galactose. Yes or no? That bond has to be break down. Who's responsible? Beta galactosidase. Then permease responsible for entering of more and more lactose should be entered. Sir, but lactose act like an inducer over here. So initially it is present. So permease is little bit what? He's allowing little bit lactose to enter already, right? And the A, that is translation or what we can say, transacetylase helps to integrate the property of beta galactosides and the permease, right? So you should know switch on, switch off model. That is very, very important. Again, I'm telling you, see, this two to evolution and genetics are the biggest vast ones. I'm trying my best to cover it, right? So look at here, we have the every gene. These are the regulatory gene and there are some structural genes. Structural genes are like Z, Y, and A. Everything is there and the function you should aware about that. Okay, so just have a look over here during these functions. Yes. Okay, so these are the functions. So how do I remember this? I understood that PIPO. What is PIPO? PIPO's head by I do remember like this. Okay. Okay, I is not inhibitor, right? Yes, exactly. That point is very much clear. So how the like operon works? Whenever there is a lactose present, only then only switch on. Please remember these things. Okay. Now the Human Genome Project. We also talk about that Human Genome Project. Its duration is this to map and sequence. Now, what is the finding? They have found 20,000 to 25 genome per DNA. Now, apart from this finding, you should know that chromosome 1 has a majority of the gene and chromosome Y has a less gene. Just you should aware about these things. An application used in the genetic testing, disease diagnosis, ethical issues are also there. What is that? Concerns over the genetic privacy, data misuse, and genetic discrimination. You should aware about the human genome project regarding the same. Now applications and we have done. Now DNA fingerprinting. So I also made the diagram table form. What is this? It is discovered by Alec. Principle based on the VNTR. What is VNTR? Again, there is a satellite also. What is VNTR? Variable number tandem repeats. Okay, tandem repeats. So they are a unique DNA sequence in the individuals. So VNTRs, these are the unique DNA sequence found in every individual, different. So DNA, what is the process? DNA is extracted, separated by gel electrophoresis. Student, remember gel electrophoresis process also there, where we're actually comparing the sample as well as the original one, right? So hybridize with the radioactive probes. That is remember that is required, right? Yes. Which gel is used in electrolysis? Gel electrophoresis. Yes. Anybody? Yes. Agar agar. Why use agar agar? Because it's have a fine pores, so DNA can be incorporated. Very good. Significance highly accurate and unique to the individual. So DNA fingerprinting is we talk about. Now let's have a rapid revision. DNA is genetic material. Watson and Crick give the model double helix. Chargaff rule you have to understood. Then DNA replication semi conservative है. किसने दिया था? Messelson star. Leading and lagging strand we talk about. 5 prime to 3 prime, 3 prime to 5 prime, you should know that. Central dogma, we talk about transcription, what is translation difference, then lack operon, and these 3 points we have talk about, right? Yes, medical forensic applications. Now, we are talking about the last chapter, that is the evolution. Okay, so when you talk about the evolution, you should aware about these two things first. Miller and Urey experiment, Oparin had a hypothesis. So this experiment is abiogenesis. Okay, what is that? Is performed by these two scientists. They have taken H2, H2, and HCH4 and H3 in such a way that they can mimic the Earth atmospheric condition which is present earlier. They have to mimic like that, right? Then Opan and Handley, what they have done? Opan and Handley hypothesis states that life originated in a primordial soup. So here is soup, right? What kind of soup over here? Organic molecules, right? I know you're hungry, that's why you are just focusing on soup. But here, the soup is organic molecules. Earth's early ocean. You know what? What we can say? जीवन का शुरुआत कहाँ से हुआ था फ्रॉम दी water ocean, right? So that is called primordial soup. Energy sources such as a UV radiation which creates this sample called biomolecules, right? So let's peep into or dive into quickly during the or in between the Miller-Urey experiment. So what is the setup? They have the methane, ammonia, hydrogen, and water vapor in such a way that they are representing the Earth's primitive atmosphere. Can you see that in the chamber? And there is electricity also there, electrons also there. To what? Yes, electrical sparks that mimic the lighting simulations. And this is continuously heating and cooling, which leads to this continuous procedure was gone through. And what they after 1 week, what they found the organic molecules such as the amino acids, glycine, alanine, and the aspartic acid. You should remember that which amino acids was found: glycine, alanine, and aspartic acid. As per the need, also this is important. The building blocks of the proteins. Okay. Now conclusion, and what are the conclusion? Organic molecules can form spontaneously under the Earth's conditions. So that is very very important. First evidence that life basic molecules we are getting from this experiment. Now, apart from that, we have evidence of evolution. First is a fossil evidence. You know that fossils, right? A transitional fossil showing the characters of both the reptiles and the birds. That is, yes, that is very, very important. That shows the fossil evidence. Then we have a comparative anatomy. Okay, we have a comparative anatomy, which talk about the homologous organs and the analogous organs. Homologous, same structure but different function. Example, human arm and a bat wings. Are they same? No, right? But they have a same structure but different function, right? For bat using for flying, human using for other things. Then analogous, different structure but similar function. Convergent evolution. See, divergent and convergent. I have the note for it, but let's me talk about convergent means different species come on the same place, same resources. Divergent means same species going away for the resources. So this is the convergent wings of the birds and the insect. They both are using for the flying purpose, right? That is the analogous organ. Then vestigial organ, human appendix, pelvic bones, and pelvic bones in a whale. Now embryonic evidence. What is embryological evidence? Similar embryonic stages. So initially, if you see the embryo of every organisms, that's shown in the picture in the NCERT, you find some common ancestry. But as they develop further, they develop some different thing. So gill slits in embryos of the fish, birds, and humans are the embryological evidence. Biochemical evidence, when you talk about, we have a DNA, proteins, metabolic pathway among the different species, right? Example, cytochrome C protein found in a many organisms, indicating evolutionary relationship. So this evidence are talk about how the organisms got evolved. Then theory of evolution, Lamarckism. See, I'm doing the quick revision. So proposed by Lamarck states that characteristics acquired during the organism's lifetime can be inherited by the offspring. But usko criticism bhi mila after Lamarckism. Acquire the trait, do not alter the genetic material. So you should aware about this much points about the Lamarckism. Then Darwinism, my favorite theory, because he's the one actually he talk about a natural selection proposed by the Charles Darwin. Example he has given industrial melanism. So before the industrial revolution, light color moth were found. Scientific name is Biston vitellaria, right? Very very common. But what happened? The lichen is a pollution indicator. क्या हुआ? Pollution बहुत ज्यादा हो गया because of the industry. Lichen तो चला गया. Once lichen is gone, but those moths require more time to adapt, right? So dark colored moth had survive advantage they got after that. Then mutation theory given by the D. Veris, okay, proposed by the Hugo Prize. You should aware about his name. Evolution occurs through the large sudden mutation that is also called saltation. They can ask you this question: what is saltation? So you should remember this mutation theory. Then very very important, that is the Hardy-Weinberg principle. For this, I'm going to use one chart. Can you see that? Yes. Everyone, please pay attention. It's hardly I'm going to take few minutes of you. So here, allele and genotype frequency, they have talk about and they give the equation. P square represents the frequency of the homozygous dominant, where Q represents the frequency of homozygous recessive. The dominant and recessive cover along with the frequency of heterozygous. So there is an example also. What is that? If the human population has a 9% people with a blue eyes, suppose 9% people are with a blue eyes, don't sing the song Blue Eyes, right? Let's focus on here. And the AA and Q square is 0.09. Then the recessive allele frequency, we can use this formula and we can get this answers, right? So this is also important. This is very, very important thing. You can they can ask for the question like this. Then genetic equilibrium. A large randomly mating population with a no evolution shows the genetic equilibrium. Means if there is no variation amongst the population, that is equilibrium, right? But that is harmful, right? So what are the factors affecting the genetic importance? First, gene flow. Please pay attention. Very quickly we can do it. Allele between the population due to the genetic variation. Yes, European genes mixing with the Native American population due to the migration, migrating people, they get married and all, right? Then genetic drift, this is very important. Random changes in the allele frequency in a small population due to the chance event. Bottleneck effect is the example over here. What is the bottleneck effect? If you draw a bottle-like structure, at the neck, it's led to low genetic variation. So mutation, sudden changes in DNA and natural selection, that is what a prepared moths evolved like industrial mechanism we have talked about, right? So let's talk about little bit about the genetic drift, founder effect, and bottleneck effect. These are very, very important things. It's hardly given in NCERT, but it's important. Random changes in allele frequency in a population due to the chance event. Founder effect. What is the founder? When one small group in from the population migrates, for example, they are getting migrated and they are populating there, then the new population leading to the reduced genetic variation, right? So that one species which is travel from common population to the diverse thing, that is called founder effects, right? Then bottleneck effect, a large population suddenly decrease. पता है इसका एग्जांपल क्या हो सकता है? इसलिए मैं एग्जांपल देता हूँ इसका कि जब लाइट होता है फॉर सर्टेन सीजन वी फाइंड मॉस्किटोज आर वेरी हाई इन numbers and suddenly they disappear. What is the reason? Bottleneck effect. Okay, even here example is cheetahs face severe population decline leading to the very low genetic variation today. So it's also bottleneck effect. One time they are very much large in quantity, but right now they are very decreased one, right? So we talk about this. Again, I'm talking about adaptive radiation and convergent evolution. We already talk about that in adaptive. What is going to happen? The process where the single ancestral species evolved, only one species is getting evolved. But in convergent, different species evolve similar traits, right? They're coming for the same resources. So species have the common ancestor in case of adaptive, but in case of convergent, they have a do not share any ancestry, right? Example we talk about in Australia. Australia marsupials evolved into the various forms such as kangaroos, koalas, right? But marsupial mammals are the example of convergent. You should aware about that. And what is the key away? Adaptive radiation leads to the diverse species, where the convergent evolution leads to the similar adaptations. Then these are the examples. See, marsupial wolf and placental wolf. What are the changes? You can see the changes. Then sugar glider and flying squirrel. See, sugar glider and the flying squirrel. What are the difference? Can you see that they are same species, but they have some adaptive things, right? That is the thing we have to understand. Now, let's talk about the last topic, that is the human evolution. And what is the important? It is in front of you. We left with a 1 minute, but we have covered all the things and we are remaining with this point. First come Dryo, then come Ramapithecus, then Australopithecus, then Homo habilis, Homo erectus, Netherlands, and the Homo sapiens. So this is the order. So what should you aware about that? You should aware about the cranial capacity and some key features that I mentioned over here, right? So when you talk about which has a larger cranial capacity, what will be your answer? Your answer will be Homo erectus, the first to use fire and advanced tool. You should aware about that. Okay, and currently, not currently, I'm talking about. If you talk about the Homo sapiens, they have 1350 cranial capacity, right? So these are the modern humans. They learn language and advanced technology. So here we have completed the whole unit of genetics and evolution. I hope you understood that very well and learn the things, right? So have you understood the last 1 minute I'm going to take? I'm not going to take more time than that. So I just want, when you're revising something, you should understand that. Whenever you're revising, you should understand that. Make a table, make a flow chart. It will be a very, very effective. So I hope you understood that very well. Okay. So I'm taking your leave. Thank you so much for paying attention. Thank you so much, everyone.